Thank you for sticking around, and thank the library for keeping us all safe. I think since it's still raining outside, and we've just been delayed an hour, that we might as well go ahead with tonight's program. So we'll make the executive decision to do that. So we'll go ahead and continue with tonight's program. We'll just be delayed about an hour or so. And so Anthony Lilienthal from the Department of Biology, he's going to talk about inclusion improves medical genomics.
Thank you all for coming and for being so patient. I hope that you enjoyed the talk and it was worth the wait. So today what I want to discuss is the first part of a two-part series in which we take a deeper dive into medical genomics and use that to inform our analyses that we use for, say, direct-to-consumer products. What can we learn from the medical genomics experiments that have been performed, and how can that help us understand, say, 23andMe and Helix to a greater extent? As we've all taken advantage of, there is a great variety of different direct-to-consumer genomic analyses products that are available on the market today. And as these have continued to develop and become much more mainstream and a much larger industry, we're finding that more and more of these companies are wanting to tell us more than simply ancestry, which is an undertaking all of its own. Now they're starting to say, for example, what kinds of traits, including those related to wellness and health, are we able to learn about considering our genomes. But with this increased scrutiny and this increased focus on health and wellness has come a host of new anxieties, if you will, when taking a look at these products. So that's part of the reason why I want to give this discussion today. I want to take a look at this topic and really dig into the medical genomics to use it as a basis to talk about platforms such as 23andMe. Now, multiple topics need to be discussed whenever we're talking about these issues. Say, for example, the ethics on reporting different disease variants, particularly when considering which disease variants we need to report. A recent paper was taking a look at one of the more recent additions to 23andMe, Bracco 1 and Bracco 2, and wondering whether or not that is something that is going to be beneficial to the general population. We need to also consider, say, what things are we willing to share with different recent research organizations as well as other companies in terms of our genetic information. As many of us have kept track of, here within the last 3 weeks, there have been meetings in which a lot of guidelines which specify those things have come up. But I really want to take a look at an issue much more central to the platform itself. I want to take a look at whether or not we can consider these genomic and medical associations relevant, and what do we need to remain cognizant of as we're going through these direct-to-consumer products. This is going to be an introduction into many of these different aspects. I'm going to take a closer view on one example set, which will then be expanded upon in the next discussion, which will be happening later this year. So the title of my focus really is that we need to have a greater inclusion of underrepresented populations in order for us to really appreciate and make sure that we're looking at relevant genomic and medical associations. My talk is going to be in three parts. First, we're going to explore the general bias which is present within medical genomics and direct-to-consumer genetic products. Moving on into the benefits that we can see, and have seen in some cases, by including these underrepresented populations. And then I want to conclude today with talking about some of the current efforts that are being performed to increase ethnic diversity, the different types that are involved, and how they are influencing this market. So, as a surprise, probably to not very many in this room, when I'm referring to a bias within this scenario, I'm referring to a specific population that is overrepresented or comparatively overrepresented in comparison to other ethnic groups when we're taking a look at both medical genomics and direct-to-consumer genomic data sets. The one that I'm referring to here is, of course, European. From this, we get a huge number of benefits. However, there are some effect, considering that a large number of underrepresented populations do not have an adequate representation in these different databases, whether it be 23andMe or things such as 1,000 genomes. Now, just to highlight a little bit of what we're taking a look at here, European populations account for 56% of the genomes which are considered reference data sets for 23andMe. whereas those that are, shall we say, in second place for representation in this scenario are Eastern Asian/Native American and Sub-Saharan Africa. Now this overrepresentation, this bias, is not something that's unique to sites such as 23andMe. This is also something that is very well noted within the genomic medical associations. Namely, say for example, if we take one example of looking at this bias for one type of test called a GWAS, or a genome-wide association study, we find that although we're seeing improvements, there is still a vast overrepresentation of European ancestry within these databases. This has improved since 2009, and considering, no matter how you consider this data set, they've looked at it in a variety of different ways, whether they're considering, say for example, the number of samples that are submitted to these different databases, or if you're taking a look at the studies that have been performed. In both of these cases, the very predominant population that's being analyzed is the European population. Now, as I said, I want to take this opportunity in order to take a little bit closer look at one of these methods, because a lot of the details which surround this method can be very informative when we're taking a look at direct-to-consumer products. So the one I'm going to highlight here is GWAS. This idea of a genome-wide association study is aimed at associating a specific region of your genome with some kind of variant, whether that be a variant which gives you a specific trait, such as increased height, or something that's associated with a medical condition. Now, the basis and the crux of this specific analysis is the variation that's present within everybody's genome. Now, when we think of the chromosomes and we think of the genome, oftentimes people will relate this in terms of genes, like say, for example, those areas of the genome which encode a specific trait that you're able to notice, hair color, eye color, etc. However, GWAS takes a different approach to this, instead looking at the variation which is present throughout the genome. So if we take a closer look at one of the chromosomes of our 23 pairs, and take a very close look at the code, dive into the chromosome, go towards the double helix, and find the code that's present within it. So in this case, we're going to be talking about, of course, the 4 bases that are forming it, A's, C's, T's, and G's. And we compare this across multiple individuals within a population. We find that even when we look at small areas of the genome, we'll have these variations in single letters of that code. These are called single nucleotide polymorphisms. Now, there are millions of these present within the genome. And in general, they're overall benign. But what's very important for this study, this type of study in particular, is that a large number of these single nucleotide polymorphisms have been characterized and localized by previous research. So now we can reap the benefits of that labor. The other thing the idea here is that a genome-wide association study is going to link one or multiple of those single nucleotide polymorphisms to some kind of trait-derived section of the genome, whether that be something that is involved with disease or other traits. Now, at its core, the idea here is to take a given population and split it into 2 sections, a case group versus a control group. The only difference that you want in between these 2 groups is going to be your trait of interest. Say, for example, you might be looking particularly at height, increased body mass index, higher blood pressure, lower blood pressure, as well as looking at specific conditions such as late onset Alzheimer's or macular degeneration. The idea here is that they'll take the genomes from these groups, apply it using the technology seen here on the right, and from that they're able to determine for hundreds of thousands of SNPs present within the genome whether or not those individuals have each SNP. The idea here is that we interrogate each SNP and find out if the individual has it. Now this probably all is sounding very, very familiar because in essence many of the direct-to-consumer products are turning this idea and turning it a little bit around. Rather than say, for example, taking it from the trait and then finding out where in the genome is associated with that trait. Instead, direct-to-consumer products are going to say, which SNPs do you have? And what traits can we tell you about from that? And because there's such a similar connection in between the two, a lot of the same problems that arise from studies such as GWASs are going to influence how we need to interpret direct-to-consumer products. Now let me take you through a hypothetical situation with a GWAS experiment. The idea here is that we've taken individuals with some given condition, let's say for example increased height or a disease such as type 2 diabetes, and they're going to interrogate hundreds of thousands of single nucleotide polymorphisms throughout the entire genome. What you want to find is a particular SNP which is considered enriched or present within your case population to a much greater degree than what is found in your control population. Found here for SNP1, each dot shown here in green is indicating an individual in that population who has the SNP. However, the dots shown here in green for SNP1 and the dots shown here in yellow for SNP2 are not particularly interesting for this specific study because it's found equally in both the case and the control population. However, when an enrichment is noted, you'll find that many more individuals in the case population, shown here in red, will have a particular SNP over the control. And so then you're able to say, okay, given what we know about that particular single nucleotide polymorphism, what can we learn about this condition? Now, before I go into a diagram which is used and usually a result of these GWAS, I want to take you a little bit through what the system that we're particularly looking at. In this case, we're going to be referring to a GWAS that was specifically targeted towards age-related macular degeneration, or AMD. This is something which is very common, especially when taking a look at European cohorts. If you're an individual with lighter colored eyes, you're likely to be at higher risk of developing age-related macular degeneration as you age. Now, this particular study looked at age-related macular degeneration within a cohort of over 30,000 individuals. They compared 16,000 patients who had been diagnosed previously with AMD and compared them to 17,000 controlled. It was a massive collaborative effort that spanned from here in the United States over to Israel and all across the globe. But the important thing to note here is that they were specifically looking at the European cohort. They were specifically looking at only people of European heritage. And this was all headed up by the International AMD Genomics Consortium. What they found is what is shown up here. Now let me take you through these results. What you're seeing up here is what's often referred to as a Manhattan plot. The idea here is this is a graphical representation of that enrichment that I referred to previously. Shown down here on the bottom is each of the different chromosomes within an individual genome. And so this is going to be comparing, again, that case and control. As we're marching down the genome, we're interrogating SNPs, saying for each one of them, is this found more often in the case population than in the control? Is this found more often in individuals with AMD over those who do not have it? As you go through, if it finds a particular enrichment, you'll see it in these peaks. The higher the peak, the more prevalent it is to the case population. Now, some of these labels probably are sounding very familiar. Let me highlight two of them. As I said, this is AMD GWAS. And so a couple of the things that are particularly strong, those that show enrichments to the highest degree, are those that are often reported on by sites such as 23andme.com. This is part of the process that companies that use direct-to-consumer genomics go through in order to give you more information. There are numerous associations that are present within this single GWAS, but only through a vetting process which not only takes a look at this study, but takes a look at various different studies studying the same issue and finding the best candidates will they arrive at some of these variations that they're going to report on to you. Now, to kind of wrap up this section in which we're talking about the medical genomic assays that are performed that are very related to direct-to-consumer products. I just want to highlight two things. First off, remember that this is a comparison. This is something in which a case and control population are being compared based around one single variant. The other thing here is that even for large studies such as this has been primarily done in Europe, in a European population. Now let's take this and flip our viewpoint a little bit. Instead of taking a look at this from a scientist who is looking at a group of individuals and saying, okay, these individuals have age-related macular degeneration? What areas of the genomes can I look into in greater details in order to learn more about this condition? Because at the end of the day, very little information is really known about what is the causal force behind AMD. And so rather than trying to trace it through a family or find the genes that way, instead they're going to find its location on the chromosome and work from there. Now let's take a look at somebody who is going to be reporting in direct-to-consumer genomics. Now, I want to take you through some data that I personally discovered in my journey through the Personal Genome Learning Center. AMD is something that I'm going to have to consider as I go into the future. Now, this is the readout that you would normally find when you take a look at 23andme.com. For those of you who are unfamiliar, let me give you a quick overview of some of the things that you're seeing here. First off, over here on the left, you have two different variants that I highlighted previously on the GWAS. In this case, the SNP is notated by these numbers here, but more often than not, it's going to be notated in a different fashion. Rather than talking about the single nucleotide change, they'll often notate it by the protein change. In this case, it just comes out as a code. Say, for example, for the chromosome 1 change for the gene called complement factor H, it's called Y402H. And then for the next variant, it's going to be A69S. Oftentimes, what they're going to show you is how many variants you have. So for myself, I get a little bit of the short end of the stick in terms of age-related macular degeneration and the SNPs, where I have both copies of the disease-related variant related to Y402H and only one copy that is present for A69S. Normally, for Y402H, you should see a T here. This is the non-disease variant. But for me, I've got both the Cs. Down here, you can see the non-disease variant of a G and a disease variant of a T. The thing that I want to emphasize here, and something that we can be informed by the fact that this is a comparison, that is in many cases, different things might conflate these issues, but at the end of the day, these are not a guarantee. These are just an indicator of greater risk of developing a particular condition. But in the interest of increasing our understanding of what's being reported to us, in the light of what we've now viewed with GWAS, I want to take us through a little bit about how they're going to report this in terms of statistically. Now, what we're seeing over here on the right side is what's called an odds ratio. This is directly informed by the comparison that was made between the case and control population. And it comes with some caveats. Say, for example, as we're walking down, going from top to bottom, we're increasingly adding more variants. Say, for example, if you have only one copy of the Y402H or the A69S, you have a relatively low odds ratio. What this means is that any number over 1 is how many times more likely to you to say, develop this. In this case, they're looking at it specifically after the age of 50. For myself, well, we got to go a ways down where we have both copies of Y402H and one copy of A69S. Down here at an odds ratio of 20.2. Large number, can be a little bit frightening. For my case, not exactly surprising. I have multiple people in my family who are, say, legally blind, and as well as having retinal conditions, something that I have to keep an eye on, but you can see here with the genomics, there's additional parts to the story. Now, the thing that this doesn't give you, as I said, this is a comparison. In some cases, like say, for example, if you're looking at specific conditions, your baseline might be incredibly small. If an individual doesn't have any of these variants, they might only have, like, say, a 1 in 1,000 chance of developing this condition. Thus, even if they had an odds ratios of 20, congratulations, you now have a 2% chance that you're likely to develop this condition. In this case, it's a little bit higher for me. For people of European descent, the base rate, the base probability that you're going to develop macular degeneration after the age of 50 is about 2 to 2.5, depending on which source you look at. Because I have an odds ratio of 20.2, well, it's a little bit more likely of a chance. We're now looking at a 40 to 50%. Now, one thing I want to emphasize is that 23andMe, as well as many of these other sites, do a very good job in conveying one aspect of research that they're coming from. And that is this idea of uncertainty in this case. What they're also going to give you is what's called a confidence interval. Roughly, oh, excuse me, I'm dropping stuff. Roughly considering, this is how confident they are in this given number here. If they were to say, I'm 95% confident in this scenario, well, they'll give you a large range. So in this case, when taking a look at my particular variant of 20, which gives you an odds ratio of 20.2, in reality, they're only saying we're fairly confident it's falling somewhere between an odds ratio of 14.4 and 28. So it could be anywhere from 30% probability upwards of 56. They do a very good job in hedging their statements in this scenario. But really, all of this information, how they have to report it, and how they come to these conclusions, like say, for example, of the odds ratio, are based in the science that it was originally derived from. This idea that a GWAS is not something that looks at baseline, doesn't take a look at a general population of individuals and say, given this condition, how many people are developing AMD? Instead, it's saying, how much more likely are you? And to be honest, that's something that can be very, very beneficial, because we're talking about humanity. This is a population that is very difficult to control all the variables. So instead, it's much easier to take a look at a group of people, separate them into 2 cohorts, and analyze them that way. Now, when we talk about this baseline, this thing is prone to flux as well. And this is where I want to branch the topic into more information about why it is important that we not only look at the European cohort. For a predominant amount of what we've shown up here, this was all talking about the European cohort. The group of Europeans who have given their data and said, yes, please use mine for research. But there's always additional factors that we need to consider when we take a look at any medical condition. Say, for example, for age-related macular degeneration alone, we have additional factors, say, that give for smoking. Diet, is particularly diets that are high in cholesterol, say, for example, things that influence blood pressure. As well as how much time do you spend out in the sun. If you're a person who works out in the sun a lot, you're going to have a higher chance. Now, all things considered, these are much smaller than the genetic predisposition, say in my case. This ranges from smoking has a general odds ratio of between 2 and 3, versus diet and sunlight exposure generally has an odds ratio much closer to 1.5 to 2. So if you're an individual who doesn't have a lot of these genetic variants, these will have a much greater effect for you. For myself, my genetics seems to be strongly contributing towards my chances of developing age-related macular degeneration. But there is a fourth thing to consider, and that is in terms of ethnicity. GWAS and studies that are associating a particular area of the genome with some kind of trait or condition are always going to be reported on and talked about in a very controlled sense. In this case, when they're reporting these odds ratios, this is only considering those S&Ps. Do you have it or you don't? But that baseline can fluctuate greatly depending upon your ethnicity. Let's compare 2 different populations. So I was telling you before, the 2 to 2.5% is what is present for European cohorts. However, if you're an individual of African descent, that baseline now is halved, and you're down to 1%. So even with just where you're from, is going to have a very large influence on how likely it is that you might develop this condition. This kind of highlights both the strength and the weakness of using this type of information in order to learn more about the association between our genome and our health. Take, for example, a benefit. In this case, because there is such a high number of individuals who are within the same ethnicity, we can make some of these determinations. One thing I didn't emphasize when I was talking about single nucleotide polymorphisms is that it's not as if every single individual in this room has the exact same of single nucleotide polymorphism sets. Everybody's going to be slightly different, and those differences can be very large when you take a look at different ethnicities. Thus, oftentimes, say for example, even those 2 S&Ps which I highlighted in terms of age-related macular degeneration, they might not even be a good diagnosis for other populations. as a hypothetical situation in this case. Further, the bias can cause us to miss some other potential signs and symptoms, or genes in this case, which associate medicine to the genome. Because at the end of the day, for both medical and direct-to-consumer genomics, this bias has arisen because of availability. Many times, historically, when these, say for example, studies were being performed, they were done in places in which a European cohort was simply the best option. Let me give you a couple of examples. Say for example, one of them is a group called Decode, and that is headed up in Iceland. The most available population for study in that case is going to be primarily European descent. Similarly, one country which has poured a large number of resources into our understanding of the genome, in particular in terms of medicine, is England. They have submitted millions of dollars to this. Again, a country which is going to have a much more readily available cohort of European descent individuals. But because we have taken so much time and energy to understand medical genomics in terms of the European population, that has hindered us in one major way. And that's this idea of how far we can extend that association. Because we've done so many of these tests, specifically within the European population, it doesn't always parse well. It doesn't always translate well when we take a look at other conditions. In fact, most times it doesn't translate well. And so, in essence, in many cases, these genomic associations can really only be used to identify disease variants within the European population. Now, what does that mean in terms of direct-to-consumer genomics? In that case, say for example in my case, I do get to receive that benefit. Well, in part. We'll get into that here in a second. But when we're taking a look at, say, some of these underrepresented populations, such as those from Asia, Native Americans, also those who are particularly from Africa and Latin America, they don't get as much benefit from this because we simply don't have that foundation yet of genetic associations to disease. This is not only found for medical genomics as well. There have been many complaints from various individuals about representation in terms for heritage, but that's going to be outside our talk for today. Now, let's talk a little bit more. We've kind of identified the bias and the problems that are inherent with it. Because of this bias, a lot of the data that we have generated, which is wonderful data which gives benefit to a huge number of people, doesn't pair well across multiple populations. But there are multiple benefits of increasing our diversity. I've been spending most of my time on the black here in these pie charts. Now let's take a little bit closer look at the non-European ancestry. Now, I told you a little bit of information in concerning, like, say, why the European population has dominated the medical genomics field. But there have been drastic improvements coming from Asia, predominantly because of that $9.2 billion investment that China is currently putting into practice for taking a look at the genome and finding out more about association between medicine and genomics. But other countries are also in this as well. Say, for example, both Singapore and Japan also have significant stakes in this market. And, thankfully, because of that, we're seeing some wonderful benefits. Say, for example, some S&Ps are now being identified, which is leading research into new areas to understand medical conditions. I'll give you a couple of examples of them. But this is really allowing us to find disease contributors that are very specific to these underrepresented populations. But, as you might expect, it also is helping us uncover some of those things that were hidden even within the European population when we were conducting these experiments. Excuse me. The first example that I want to highlight here is going to be related to cleft lip and palate. Of course, something that is influencing many of the young population today, something that is has significant visibility within various different organizations. But the thing I want to highlight here, in terms of why it's so important for us to incorporate some more diverse populations, is this idea of who is the most affected by this condition. When you take a look and compare the rates of cleft lip and palate present in children across the world, Asia and those who are here native to the Americas have a much higher rate of cleft lip and palate conditions, while Africa is the lowest rate at 1 to 2,500 births will have a cleft lip and palate. This highlights how much better of a system of analysis looking at Asian and Native American genomes would be. Because with that higher prevalence, they likely have higher numbers of single nucleotide polymorphisms and disease variants, which we can learn more about this condition through. Let me give you an example. Now, I work here at the University of Iowa in the Manic Lab, and I was part of a project which did just this. We took a look at a Filipino population and compared it to a European population. By exploring the genomes from these Filipino donors, we were able to isolate a new gene, which was involved with the formation of cleft lip and palate called ISMN1. The important thing to note here, though, is that this association would not have been possible if we had only taken a look at the European cohort. It simply wasn't present, or at least statistically speaking, not to a significant degree. But it's not only the Asian population that is in need of some increased scrutiny. That was one of the benefits. There are many others which they're starting to discover thanks to the added support that is coming from countries such as China, Japan, and Singapore. But another area that really needs some desperate attention is, say, for example, Latin America and Africa. If we take a look at those in respect to another condition, in this case asthma, we have what is often referred to as the missing patients. What happens within these populations is that children from Puerto Rican specifically and Black heritage have a much higher incidence rate of asthma. And not only that, oftentimes current medications that are used to help treat asthma and to alleviate its symptoms are not necessarily effective for these populations. These are some of the consequences of that European bias. Because we've only looked at it through that lens, we've missed some possibilities. Thankfully, because individuals have noted this increased prevalence of asthma within these groups of children, we've made some strides in that direction. In a study of 772 people, a variant within the gene interleukin-4 was found to be associated with higher asthma rates, specifically within African American children. This was done by a study back in 2014. Now, this is that first step in the process in order to understand the condition to a much greater degree, as well as help to develop better medicine specifically for these populations. Now, I've been taking a look at this through a very specific lens, representing underrepresented populations in order to help them with their specific needs. Oftentimes, there are specific variants that you'll only find in those populations. However, when you take a look at these specific populations, we can also learn more about populations we already know about, namely European. The final study that I'm going to highlight today, we're going to go to Greenland. We're going to take a look at the results from a Greenlandic Inuit study, which was studying specifically height in this case. So a little bit of a step away from medicine, but this is too perfect of an example not to take advantage of. When analyzing 191 Greenlandic Inuit populations, they found that there were several SNPs which tied genes that were responsible for their ability to metabolize fats, in this case, with their height. Now, this makes sense. As I've been emphasizing with multiple of these populations, we've been taking a look and saying, this is something that's much more present within that population when we took a look at African American students, as well as taking a look at cleft lip and palate within Asian children. But in terms of Inuit populations, they have had a high-fat diet for numerous generations. And so this means that they're very likely they have also made some adjustments and some changes in their genome, which have made it so they're better able to do that. And it's been studied in multiple cases. Now, what's most important to note here is that this is not something that only informed us about the Inuit genome. It was found that through follow-up studies, these same genes were influential within European populations. Again, this was something that, although the GWAS was done correctly, because of its prevalence within the European population, it simply wasn't statistically significant enough for somebody to highlight it and take a closer look at it. But thanks to looking outside of the European population, we learn more about ourselves. All right. Now, for the final part of my presentation today, I do want to highlight some of these efforts that have been taking place. Something, by returning to this slide, I want to highlight. Notice that back in 2016, GWAS studies were still very predominantly European. We were talking 81% of the population were, 81% of the samples were from European populations. However, when the market has opened up, we're starting to see this bias start to dissipate. Say, for example, in this case, for 23andMe, 56% of the population who have submitted it and have been considered for reference have been from the European, with greater amounts in sub-Saharan Africa and Native American. One example of a project that has helped work in this direction is the African Genetics Project. This one added 1,000 African individuals to the reference genomes for specifically 23andMe between the years of 2016 and 2018. Excuse me. This is truly a feat because it is extremely difficult to find these reference populations, specifically within African descent. Because in order for these to be considered, only people whose grandparents, all four grandparents, were born in the same country in Africa can be considered for this baseline. The idea here is that Africa, as the place that Homo sapiens kind of migrated from, has a very diverse genome. Over the entire continent, there are multiple different ethnicities that need to be considered. That's why oftentimes we see the breakdown of it. As such, in order for them to be considered in these cases, we need to take populations from small areas and analyze them individually in order to find these unique variants. Some other projects that I do want to highlight here, if you're curious about other groups that are increasing the diversity of medical genomics, as well as 23andMe and other direct-to-consumer products, are going to be Ancestry.com's Ancestry Human Diversity Project, the Global Genetics Project, again from 23andMe, Human Heredity and Health in Africa, H3 Africa, as well as study of Latinos or Hispanic community health studies. Now, all four of these highlight an organization specifically looking to increase an underrepresented population, but I don't want to misinform here, because there have been numerous efforts from companies and from individuals which are also helping us understand these genomes even better. Say, for example, we have multiple research groups that are very focused on specific conditions. Say, for example, the one I gave here at the very beginning of the International AMD Genomics Consortium. And say, for example, in Japan, the Garden Health, which has received recent support from the Japanese bank in order to take a further look at the genomics of specifically cancer. And this is not even to highlight the numerous individuals and labs which are working with these companies specifically in order to understand the genome better. All right. In conclusion, I hope that I've kind of conveyed to you a little bit of the both sides of this coin and how our understanding of medical genomics can really inform and help us understand some of the results that we're getting back from 23andMe a little bit better. A lot of this previous work was done in European cohorts, in groups that were primarily European, and has led to numerous medical associations which are extremely relevant. However, many of them cannot be taken out of context. As such, we need to increase the inclusion of underrepresented groups in order to help us understand not only these underrepresented groups, but also further understand our own genome. With that, I would like to give a huge thank you to the Personal Genome Learning Center and Dr. McAllister for allowing me to take a little bit of a deeper dive into this topic, as well as all of you for sticking out the weather, sticking out the rain, being patient through it all, and allowing me to present some of my findings before you today. A little bit of a plug here at the very end. I do recommend next month's talk. Another student is going to go through a different method in order that we can take the entire genome and learn from that to generate what's called a polygenic risk score. In many ways, I've been focusing on, say, okay, how do these single changes within the genome, each individually, increase our risk or increase our chances of having a specific trait. But in this case, it's taken a much more holistic approach, taking the entire genome, in order to learn more about those traits. With that, I'll take any questions you may have.
Thanks, Anthony. And if you have questions, raise your hand and I'll bring the microphone over to you so that we can get your question recorded.
Excuse me.
Thank you for such a great presentation. Are there people without either of those two genetic markers that develop macular degeneration?
Yes. And that really emphasizes that these really are just a single look. Not only are there just people outside of that group, there may be other variants that are associated with that gene. If we look back at that Manhattan plot, we're not going to do it in that respect. Here, hold on. That will take forever. Each dot you're seeing here along this peak for, say, for example, CFH, is one S&P. There are a large number of single nucleotide polymorphisms that are considered with this gene. And even if you don't have those genetic predispositions, they can also develop age-related macular degeneration.
Because my
Go ahead.
follow-up question is just that people with the higher concentration of the genes might not develop macular degeneration, right?
Very true. It's just a metric that says if you have this, you have a higher chance, a higher risk. There are additional factors that need to be considered here, including lifestyle, as well as even if you had the worst lifestyle for it, as well as all 4 variants that were present for this highlighting macular degeneration, you might still not develop it. You're welcome.
You guys are well behaved tonight.
Thank you.
So I certainly understand your examples about the generalizability of the data based upon where the data came from. And I have not done 23andMe, so I don't really know this, but since all 4 of my grandparents are European, I would like my comparisons and my odds ratios and confidence intervals to come from a population that represents my genomics, because I would think that would be most specific. Mm-hmm. But what do you get from 23? You showed that how their population database has changed in percentage. So if I get my results, are they going to compare me just to everybody or just to my own European background?
Within this context, that's kind of the beauty of these types of analyses. They're going to compare you to everyone in these cases. Although they might have additional information that says, if you are not of this population, this might not be effective to you. I'm taking my response here from another gene, which is oftentimes taken a look at, which is BRCA1 and BRCA2. Those are very specifically geared towards a couple of variants that come from a specific population of Ashkenazi Jewish heritage. And so even if you're outside that population, those might not be as influential to you. Does that answer your question?
Yeah. So 23andMe compares you to whoever is in their database at that
Everybody
point
within
in
their
time.
database, exactly.
But I would just add on that the reports are ultimately going to be shaped by the ethnicity from which the science is obtained. And so, for instance, on age-related macular degeneration, there's sort of a caveat explanation of this. These predictions are based on individuals of European ancestry, and if you're not of European ancestry, these may not be as applicable to you with that different ancestry.
Similar to that idea of the baseline is different based upon ethnicities. This is just taking a look at 2 variants. There are additional genomic factors which do contribute to it based upon which population you're from.
So the common disease conditions that they report upon, which ones have the smallest confidence intervals?
Ooh, tough question. Okay. Which disease populations have the smallest confidence intervals in terms of, say, the odds ratios?
It would be the GWAS whatever has the best GWAS sample size.
The better the association, the
and
smaller the
more
confidence, right?
heritable trait. So I don't, like, if you wanted to know, you could look at, they rely upon an underlying GWAS. So the bigger the sample size there, the better the predictions will be. And then also how genetic a trait is. So Anthony mentioned that, you know, there are environmental factors in AMD. So if those play a bigger role, then your confidence intervals will be larger because the influence of genetics on your outcome is less important.
I understand that, but I was just trying to figure out which diseases do they report upon that are going to give you the most, can you have the most confidence in?
Lactose intolerance, they're really good at that one. But that's not a GWAS.
Thank you.
I believe there's a question over here. Oh, yes?
Yes, I'm just wondering whether 23andMe or any of the other companies, it does research on Parkinson's gene or predisposition for that?
Yes, though I'm not very familiar with the specific research at this time.
23andMe has a Parkinson's prediction, and so they have a report. And part of what went in development of that report was a collaboration with the Michael J. Fox Foundation. And so they recruited a lot of Parkinson's patients into their database as part of that research effort as well.
Thank you.
So I was just going to mention that one point that I don't think came through,
Okay.
and maybe you didn't intend to make it, but the fact that you're talking about increasing the diversity, that doesn't mean that you can lump all of these diverse populations together to do the genome-wide analyses.
No, you're exactly right.
Right, so they still have to be population-specific and adjusted for background variation in some cases too, to identify these candidate risk genes or risk variants.
Exactly correct. As I said, each ethnicity is going to have its own set of S&Ps. Thus, if you were to try to combine them, many times those nice, clear peaks that we're seeing here in a Manhattan plot would be diminished. The best way to do it is always to use the control, which, and frankly, the only way that you would want to do this is that you would want to have the control population which has the most homogeneous, what we call background genetics, or say ethnicity in this case, that is related to your case populations. And so you would have to compare between multiple studies. Say, for example, what I was highlighting previously with my work in the Manic Lab. We didn't combine the European and the Filipino cohorts in order to find ISMN1 as a gene. Instead, we took a look at Filipinos, compared individuals of Filipino descent who had cleft lip and palate to individuals who were Filipino who did not, and that's how we were able to arrive at it.
So for some of these populations, obviously, you'll never have the really huge sample sizes that the European analyses have had.
Well, with continued support, we'll have to see, but unlikely for some populations, that's for sure.
Will you talk a bit about, one of your slides said publishing information about Bracco can do more harm than good. Can you talk about
Sure.
why that is?
I'll tell you a little bit about that. So the specific anxieties that were related from that particular article, and the title of that one being Bracco might do more harm than good, as you said, was that it's misrepresenting a lot of the population that you're viewing. BRCA1 and BRCA2 are just two SNPs which are related to an increased risk of developing breast cancer. So their main concerns there are that, one, it doesn't include all of the different variants which have been identified within this respect. Only two of the most, shall we say, analyzed ones. Say, for example, because of the prevalence of the Ashkenazi Jewish population studies that were present that helped identify them. And thus, if you had these conditions, yes, it's helpful to you, but, let me rephrase that. If you have these particular SNPs, yes, it's beneficial to you. But even if you don't have them, it really doesn't give you the full picture of whether or not you have a higher chance of having breast cancer. That was their main concern in that particular article.
Well, I was just going to say that I've had a DNA test from 23andMe, and one of the things you get from it is what, they really pinpoint what country your ancestor came from. So it would seem logical to me, then they probably do that first and then compare you to that group. Possibly, I don't know.
Possibly, in these cases. For some other populations, they don't really have the benefit of a very pinpoint accurate one there. Say, for example, I've heard, and when I was looking through this research and trying to get, say, a feel for a lot of those hang-ups that people are having for these particular companies and products, is the fact that there's such a broad bins that they're placed in, say, for example, particularly African populations and African-American populations.
Whenever you registered your sample in 23andMe, as well as they ask you if you know where your grandparents were born, and so that's also part of what informs how they treat your sample is the birthplace of your grandparents that you put in, if you know that information.
So what you said about the BRCA 1 and 2, don't you think that's true about a lot of the conditions?
It is.
Yeah, so that's always been my problem with the 23andMe and any sort of direct-to-consumer testing, is that you could talk to cardiologists here, and they're not going to say that you don't have any risk for heart disease if you don't have, this variant, and that variant.
I like that you specifically mentioned heart disease, because some variants that are specifically not reported by many of these things, say for example, are very specific to given populations. Say for example, one of them that was most recent is that for African-American, African populations, the fact that they have a particular variant within their genome makes them more, one, predisposed to heart complications, as well as less likely to be effectively treated with specific medications. So you're absolutely right. The idea here is that we have to take these comparisons in mind, because we're not getting the full picture here.
But do the consumers know that? I mean, that's the problem. We need more counselors and that sort of thing, and more training of physicians and, medical personnel.
That's one side of it.
In order to be able to.
And that's one side of it, and I definitely agree, is that there has to be a greater look into this. And so really, at the end of the day, when we're using these medical associations, especially those that are coming from, say, 23andMe, these are just, say, little vignettes, little things that are going to be, say, if you have it, cool, now you have a little bit more information. If you don't, well, there's a plethora of other research out there which can help you become more informed. But as we'll find out next month as well, there are additional types of tools that people are starting to develop to try to account for this gap in between the relationship between SNPs and these conditions.
Well, and one thing that, I wouldn't say it's emphasized on 23andMe website overly. They oftentimes say, there are other risk factors besides genetics, but one sort of real indicator is if you have a family history. And so if there is a family history, and then if you test that you do not have the variants, that's very false confidence that you're not at risk. And the FDA was very concerned about this in terms of breast cancer,
yes.
Of finding out that you're not a carrier of those variants, but you have a family history of breast cancer. You should still go be active in mammograms and screening. And so,
yeah, as a couple of people have touched upon, and I'm a little cynical that there's a lot of innumeracy around that the general population is not very good on subjects of probability and risk analysis and so on. And yeah, guilty. But the important thing that I think often people err on is that the absence of a particular marker or so on does not prove that the risk is not there. That they understand that if you've got a risk marker, well, then you have some risk. But the danger is saying that, oh, I don't have those, so I don't need to worry about that. Exactly. And that, I think, is a big danger in that we shouldn't shut this down, but we need to help people understand what it doesn't do as well as what it does do.
Exactly, and that's what a lot of those say. For example, that article that we were discussing, that's exactly the point that they're getting to as well. Because you don't want to give people false information. You don't want to give them a false sense of security. It's still very necessary, going back to Brocca, say, for example, that regular tests are conducted to make sure that that's the case.
Well, I thank you again for coming, and hope to see you again at our September meeting, and we'll start sharp at 6 o'clock, hopefully. And
Hopefully.
hopefully we won't have one of these spring rainstorms.